All right, I think we are there, and I think we'll start on time. Um, is it 10:30 on your clock, Bonnie?
It is.
Okay. So, welcome to EPP seminar today. Maybe I can put on my camera. I don't know if that's a good idea. Looking pretty sweet. It's colder than it looks out. We have an invited speaker today that Bonnie works with from Bess. And I'm just going to first ask for announcements. Just turn on your mic, blare it out. Anyone? I'm going to do my same announcement since I got nice feedback from the last speaker with the, I think, total of 5 feedback forms. So unless you're sending them directly, which is wonderful, I'm going to just have to call you all out and say that the students aren't really filling them out. And I do think that the speakers appreciate it. And I'm just going to sort of beg you one more time, take the 1 to 2 to 5 minutes, fill it out, send it to me. I populated folder and then send it to the speaker. Robbie, I don't know if you'd like that for today or not as your student. These are just feedback meant to be constructive. You can just let me know. I got to think it's helpful. So what do you think of that, Robbie?
It would be great, but I don't want to have you an extra effort if it's
no. This is what the students are supposed to do.
Okay.
I'm just not like a policeman, so I just beg and I don't come around with a club or anything. But if I do get some, which I think again I plead with you all, I think it's worthwhile. I think the speaker can learn a few things. If there's some things you can be constructive about as far as how to help them be more, have more clarity or slides that really gummed you up, I think that's just super helpful. Okay, announcements, 3, 2, 1, bong. So now I'm going to turn it over to Dr. Bonnie Only and let her introduce our speaker. And thank you all for coming.
Okay, thank you, Kurt. Good morning, everyone. Our speaker today is Ravi Teja Kumar Nilapalli. Ravi grew up in a rural part of southern India, and his father is a 5th-generation farmer. His family has a 30-acre land holding in India where they grow rice, corn, peanuts, and cotton. Ravi completed his bachelor's degree in agriculture in India and his master's degree in plant and soil science at Texas A&M. And while at Texas A&M, he worked on projects involving water management in rice, mushroom cultivation, and sorghum breeding. During his master's degree, he also studied the effects of co-inoculating endophytic fungi and bacterial nitrogen fixers in peanuts, which has relevance to his topic today. Currently, he is a doctoral student in biosystems engineering and soil science, and Dr. Sindhu Jagadama is his advisor. So, on a personal note, Ravi shared that he enjoys digital photography and watching tech videos. He can play 6 different sports and represented his undergraduate university at the national level in 4 of these sports. An interesting fact about him is that he can recite the alphabet from Z to A in less than 3 seconds. So back to our topic today. Ravi's doctoral project actually involves something very different, a rotation of organic grain crops. But for today's seminar, he's going to share his findings on a side project that is not part of his doctoral work. This project was inspired by work he did with an organic farmer in India, and he received a faculty student grant from the graduate school to support it. Okay, Ravi, the floor is yours.
Wow. Thank you very much, Dr. Only, for that wonderful introduction. And also thank you for the invitation. So, coming from a farming background, I have seen many farmers prepare these bio-formulations in India, and I was dazzled by the effects that these inoculations have on the plant growth. So, thanks to the SFRA grant, I was able to collaborate with Dr. Only to conduct this study as a mini project. And I prepared 2 different bioformulations and tested them on the plant growth characteristics of corn, soybean, and wheat. And in today's talk, I'll be presenting you with the findings from our study and conclude by showing the functional significance of the microbes that are present in these organic bioformulations. Let's dig into some background. So, a bioformulation is something that consists of biologically active substances, and that can be living microbes themselves or their products such as spores and metabolites. And there are many such bioformulations in the market right now, and they're used for plant growth promotion, nutrient acquisition, and disease control. However, in India, majority of these inoculations that are used by the farmers are prepared by the farmers themselves locally in their farms using cow manure and some of the plant extracts from neem, calotropis, which is also called as milkweed, and the pepper, garlic, marigold, and tobacco. So these bioformulations, they go with different names from region to region, but in this picture, you can see a farmer who is preparing Jeevamrutham that is used for plant growth enhancement. And this is one of the inoculations that I replicated for my study, and I termed it as a probiotic inoculation. And another such bioformulation with the same ingredients is the Beejamrutham that I used for this study, and it is used for seed treatment. I couldn't come up with a fancy name for this bioformulation, so I'm just calling it as a seed treatment bioformulation. And there hasn't been much research on these bioformulations that are prepared by the farmers themselves back in India or here in the West, but something close to these bioformulations are biodynamic formulations. These are again prepared using biological practices, but they also include some of the spiritual techniques for their preparation. And a major product that is widely known in US is a biodynamic compost starter. It was prepared by Dr. Pfeiffer in the early 1940s, and everything about these biodynamic inoculations and their preparations were nicely summarized in this paper called Biodynamic Farming and Compost Preparation by Steve Diver in 1999. So, why do we have to study them? Usually, any inoculation that we introduce has to benefit the farmer's ultimate goal of increasing productivity, right? But for a microbial inoculation that a farmer is preparing by himself in the field, we do not know what is the microbial composition of that bioformulation. And we do not know which are the microbes that are surviving once they are inoculated into the soil. And we also do not know how these inoculated species are interacting with the native soil microbes, and what are the functions that these microbes can contribute, or how they interact with the plants. And we also do not know what they can or how they are able to impact the plant growth in an already established plant, soil, and microbe ecosystem that is present in that particular piece of land. Therefore, in this study, we wanted to find if these bioformulations can really enhance the plant growth and understand the functionality of the bacterial and fungal diversity in the soil after inoculating with these bioformulations. So, the important part of this project was to prepare the inoculation itself. And here are the ingredients for the seed treatment inoculation and probiotic inoculation. So, we used cow dung, cow urine, lime, and the soil from where we collected the cow dung. So we collected the cow dung from the Caney Fork Farm in Middle Tennessee. They had this organic ranch, and this cow dung is from uh, freely fed cows, black Angus cows. And we also used the soil from the same field because it was organic and it was under natural farming conditions. And for the seed treatment inoculation, I mixed all of these, and I used lime to increase the pH. That way, I can inhibit any pathogenic microbes that can be troubling the seed germination in an immediate through an immediate application. So this was just fermented for 2 days, whereas the probiotic inoculation, though I used some of the similar ingredients, I used cow dung, cow urine, and then the um organic field soil again. But this inoculation was fermented for 7 days. So, in order to feed the microbes and for them to grow in the inoculation, I used brown sugar and the pulses powder to act as a source of nutrients for the microbes that are increasing in that 7 days period. And we conducted a lab germination study on corn, soybean, wheat after seed treatment with the seed treatment formulation, then followed by greenhouse study to look at the germination and seedling vigor. So seedling vigor is nothing but a product of germination percentage and the overall seedling length. I used the germination percentage in each pot, and then I actually seeded some extra seeds. If I had to grow one soybean and corn plant, I seeded 3 plants, and then I carefully removed the other 2 and then looked at their seedling length. So seedling vigor was calculated by the product of germination percentage in each pot multiplied with the seedling length of the crops in that pot. Later, followed by a greenhouse study uh, to look at the multiple soil and plant growth parameters up to 30 days. And for this purpose, I filled the pots with the soil from the ETREC Organic Grain Crops unit, where I have my major project on organic grain rotational systems. And I arranged the pots in a randomized block design, and we had 3 different treatments. The first treatment was a co-inoculated treatment, so in which I initially treated the seeds with the seed treatment bioformulation, and then applied with the probiotic formulation on the 7th and 15th days after sowing at the rate of 4 and 10 ml respectively. And then the second treatment was only provided with the probiotic inoculation. And then the third treatment was controlled with no inoculations. And the data was collected on 7, 15, and 30 days after planting. And I used next-gen sequencing to classify and characterize the functionality of the bacterial and fungal diversity. So the samples themselves included the raw materials like cow dung and cow urine, and the soils from the ETRC soil and the canifork soil, and the microbial formulations that were prepared, the 2 inoculations, and the soil samples that were collected from the rhizosphere zone on 7th, 15th, and 30th days after planting. Then the DNA was extracted using the Kyogen Power Soil Kit and validated on the gel, and we amplified the 16S and ITS regions to through the primary PCR, and then we indexed them with the index PCR. Later, the PCR product was quantified on the nanodrop, and then we loaded them onto the MySeq at 4 picomolar concentrations. The OTUs were generated, analyzed, and classified using the R program. And some other details I would like to mention here was we used the RDP tool to for the taxon assignment of the bacterial and fungal OTUs. And then we use the FunGil to assign functional types to the fungi. And the primers that I used for the bacteria were 341F and 785R for the 16S region, but whereas for the ITS2 gene region, we used a custom mixture that was provided from the Greger and Doe experiment in 2020. So coming to the results, Let's see what are the abundant families in the inoculations compared to the raw materials themselves. And this is the relative abundance bar chart for bacteria. The families that are highlighted here are lactic acid producing bacteria, the Enterococcaceae, Enterococcaceae, Streptococcaceae. These are all lactic acid producing bacteria. And there are studies showing that the effect of lactic acid bacteria in plant growth promotion, pathogen suppression, and the production of indole acetic acid. So these might actually help the initial germination of wheat seeds that I'll be showing you later on. And whenever you try to do something good, it's not always good, right? So there's also some bad that should happen. So along with that lactic acid bacteria, we had some saprophytic bacteria, but they also contain some of the pathogenic species to the plant. And majorly in the cow urine, you can see that the Pseudomonadaceae, and it didn't come up much in the probiotic formulation, but it was slightly there in the seed treatment bioformulation. And the Moraxylaceae, this is mostly saprophytic, but again, it also has some of the fun bacteria that help in nutrient cycling, but again, they also contain some of the pathogens within their family. And looking at the fungal abundance, we can clearly say that one family is dominating here, the Neocalistigmataceae. So this is mostly a symbiont in the digestive tracts of the cow, and this was majorly represented in the cow dung and then the both inoculations themselves. And the rest of them are all saprophytes, and not many of their functions are known yet. And some of the families that I can identify and say here are Hypocreaceae. So this is the family that contains the trichoderma, and we know that trichoderma has many uses, and it is commercially used as a biocontrol agent in many of the crops. And we also have the Pleosphoraceae. So these are actually, both of them are present in the cow urine. We have the Hypocreaceae and Pleosphoraceae. Pleosphoraceae, it contains the alternaria species. I just wonder how come a biocontrol agent such as trichoderma or the alternaria will be surviving in the cow urine when they're coming. And some of these are again thermophilic, that can sustain in the harsh conditions. And if a farmer, if he's trying to apply these bioformulations in a arid zone or in a tropical and temperate areas, he need not worry about the temperatures. And let's look at the effect on plant growth. Unfortunately, we did not see any effect of these bioformulations on the growth of corn and soybean. I also looked at some of the soil properties such as the microbial biomass, carbon, and pH. Again, there is no effect on those soil properties. However, the wheat plants, they were very responsive. As you can see here, this one is the seed treatment inoculated uh, treatment, and this was the control. You can see that the seeds that were treated with the seed treatment inoculation were growing considerably better. Hence, I would like to focus on the wheat data from here onwards, since we saw some significant effect them on the wheat. And if you look at the seedling vigor, it was again significantly different from the control. And along with that, we were also able to see the effect of the co-inoculated treatment on the tiller count, shoot biomass, and root biomass of the wheat plants. So the treatment with the probiotic formulation alone, it was doing slightly better compared to the control, but it was not a significant difference to be mentioned. But yeah, the wheat showed immediate response, and seedling vigor and the other plant growth characteristics were uh, better up to 30 days after treatment. So after seeing this significant effect on the plant growth of wheat, we looked at the microbial composition of the soil, hoping that it was something that was related to the microbes. And as shown here, after 30 days, both the bacterial and fungal composition varied between the treatments. The red box represents the composition of the co-inoculated treatment, and second one is control. Uh, and in fact, we did a multivariate analysis, and it showed that the microbial composition actually varied across all the treatments between all crops at all the sampling times. Knowing that the composition was really affected between the treatments, I looked at the major abundant families in bacteria and fungi, and here this chart represents the bacterial abundance, including the raw materials as well as the soil samples. So the first bar here, it represents the cow dung, the cow urine. And this ETRC soil is the one that I used in the pots. This is the soil, the coniferous soil. This was used for the inoculation preparation. This is the seed treatment formulation, and this is the probiotic formulation. This is the wheat soil that was co-inoculated after 30 days, and this is the wheat soil that was the control at 30 days. And you can see that the Pseudomonasaceae, this was the highly abundant, mostly represented family in the bacteria, and it was mostly found in the cow urine, ETRC soil, and coniferous soil. So this actually again contains the saprophytes, but also some of the pathogenic species. And I was glad that this was not majorly abundant in the uh, bio-formulations that we prepared. Then, apart from that, the other major families that were represented in the inoculations as well as in the overall soils were GP6, Bacillinaceae, Bradyrhizobiaceae, GP4, and Chytophageaceae. So all these are again saprophytes, but they are some of the robust bacteria that can cycle the organic matter, stimulate growth, and suppress pathogens, as well as some of them help in nitrogen fixation and phosphorus solubilization. Then I also looked at the fungal abundance in the same treatments as before. We have the raw materials here, the inoculations, and then the co-inoculated soil, and as well as the control soil in wheat. So the first thing that we can observe here that was different from previous thing was the Trichosporinaceae that was present in the wheat co-inoculated soil in the co-inoculated treatment. So this family is majorly saprobic in nature, and it produces lipids and biochemicals. One function that I could find regarding that was it was commercially used for biofuel production, but again, no major function that was identified when it is in the soil. And the other families include the Cucurbitariaceae, Motorelaceae, Trichomaceae. These are all again mostly saprobiotic fungi, but their functional traits are not yet known. Apart from them, two other major families were the Nectariaceae and Rhizopodaceae. So this Nectariaceae and Rhizopodaceae, these are saprophytes, but they contain some pathogenic species. For example, Nectariaceae, it hosts Fusarium species, and Rhizopodaceae hosts Rhizopus species. So we can say that, okay, these are some of the pathogenic families, and they are highly abundant in the ETRC soil, the soil that I used in the pots. So maybe that was the reason why we were not seeing good yields in our organic field trials. Then these are also present in the co-inoculated treatment, but they were very abundant in the control soil compared to the co-inoculated treatment. As you can see here, both the families represented very well in the control compared to the co-inoculated treatment. So we were able to see some suppression there. So let's look at the functional traits of the whole fungal diversity, because I could only do it for this one. the time period that I had was like 6 months, so I could only learn it for the fungi. I heard that we can even do it for the bacteria, but it's really complicated. So, looking at the relative abundance graph of the fungal abundance based on their functional traits, I looked at the inoculations. These two are the inoculation seed treatment and the probiotic. We have the field soil as well as I differentiated the soils at different crop stages. So we have the one at 7 days, 15 days, and 30 days. So overall, we could see that there is an increase in soil saprophytes, and this increase in soil saprophytes is actually helping in the suppression of pathogens. So as the days goes on, as the soil saprophytes increase, we were seeing a decrease in the uh, plant pathogens or the families, and this might have actually contributed to the better plant performance in wheat, even though we couldn't see anything in the corn and soybean. And this might have actually boosted the plant vigor initially, and we were able to uh, see that enhanced plant growth in the wheat. And it is to be concluded that these bioformulations enhance the growth in wheat, and the microbes that are present in them had various functional traits such as plant growth promotion, nutrient cycling, nitrogen fixation, and pathogen suppression also. But I think that the application of these bioformulations periodically can help in maintaining the balance of the microbial diversity, because I've seen farmers in India apply these every 15 days to the soil as well as onto the plants. And I mean, if we think that the current farming practices, such as the tillage, fertilizer, pesticides, or even the climate, they are favoring the survival of some of the microbial species. Say, if tillage, you are breaking the aggregates, and maybe only the aerobic bacteria can survive. And the fertilizers and pesticides, they might be killing some of them. But if there's a heavy rain and flood, maybe only the anaerobic bacteria are being favored, and any aerobic bacteria might die. So all these practices, I mean to say, they might be favoring either the pathogens or sometimes even the good microbes too. But if we can continuously rejuvenate the soil with these bioformulations, we can actually mimic the nature to some extent, and achieve the so-called sustainability in the farming sector. And I would like to thank the Kenny Folk Farms for providing us with the manure and support, and then the SFRA grant from the University of Tennessee, and Miss Brown and Emily Walker for their help.
I want to express my gratitude to Dr. Jagadamma, who has gone above and beyond to help me, as well as to Dr. Onley and Dr. Oliver and Wilson, who have worked with me and contributed significantly.